speaker is Lena Dorfschmidt from the University of Philadelphia, and her title is Development of Cortical and Subcortical Asymmetry from Mild Gestation to Old Age.
How do I get the pointer on the screen? Hi, I'm Lena Dorfschmidt from the University of Pennsylvania, and my work is motivated by the idea that asymmetries are a fundamental principle of brain organization. We know that asymmetries are present in the prenatal period and at birth, and that they change fundamentally over the course of life. We know much less, though, currently about the full lifespan development of asymmetry. Recent work has also shown that there are strong differences in neuropsychiatric and neurodegenerative disorders on the left and right hemisphere. Now, we also know, lastly, that there's strong variability in asymmetry between individuals. To address these concerns about modeling asymmetry, we propose a normative modeling framework to trace asymmetry over the course of life. So this is akin to the World Health Organization's brain growth charts. Growth charts. We're proposing the brain ones. Pediatric growth charts track children's height and weight and allow to map an individual against the normative referencing standard. This allows us to detect early deviations and intervene in case of atypical development. Previous work from our group in a sample of 100,000 scans has recently highlighted the potential of normative referencing standards to quantify individual deviations from global and bilaterally averaged neuroimaging metrics over the course of life. And these trajectories are not only useful for tracking the mean of population development, but also variance. If my pointer worked, I'd point at the normative variance plot right now. So we can also contrast the different phenotypes that we model, and this helps us to understand periods of vulnerability and opportunity for intervention. In my own work now, I use an even larger sample of 174,000 scans from 128 studies spanning the entire lifespan, including prenatal development. And we've extended the framework now to be regionally specific, so we have left-right separate models of brain development in 4 imaging phenotypes. So the models we use are generalized additive models of location, shape, and scale, gamma-SS. We model the development of age in neuroimaging phenotypes while correcting for sex and study effects. And these models allow us to correct each study or normalize each study, not only in terms of their mean, but also their variance and skewness, which in turn means that we get more accurate centile scores or subject-specific deviations from the norm. Having estimated regional trajectories in the left and right hemisphere for gray matter volume, surface area, cortical thickness, and subcortical volume, we first characterized the milestones of development for each trajectory. So we estimated the peak of a trajectory, the peak rate of growth or development, and the peak rate of decline or deterioration. I'm illustrating here now the age at peak for each of the phenotypes. And as you can see, there's substantial variation both between phenotypes but also between hemispheres. Now, we were interested in characterizing the complex interplay between these milestones of development. And to this end, we've mapped it onto this 3D color grid. I'm really sorry that the pointer isn't working. Where we map each milestone onto one axis and color it. So you could imagine a region growing very early and declining very late. We would call this first in, last out. It's a well-known concept in the literature. It characterizes regions that develop early and are thereby also resistant to decline or deterioration later in life. You could imagine in pinkish colors the opposite, last in, first out, so regions that develop late and then decline quickly. These are two illustrative trajectories I'm showing here right now. As an example, for cortical thickness, indeed, we observe a pattern of these first in, last out across the cortex. Most of the cortex is yellowish, in particular somatosensory regions, for example, where we see some last in, first out, or pink regions in frontal areas. Lastly, our models also allow us to estimate subject-specific deviations or centile scores. And using these, we can characterize individuals from patient groups against normative controls. We now have, given our sample size, case controls groups of in the thousands. I'm illustrating here for the first time with left-right specificity case control effects in a number of disorders, where blue indicates patients having decreased centile scores compared to controls, which is true for most of the regions and phenotypes I'm showing you here right now. Now, I started by saying that I'm actually interested in asymmetry. Using these maps from the previous slide, we can characterize the hemispheric differences in case control effects. So correlating the left and right hemispheric case control maps from the previous slide, we see substantial variation in the interhemispheric correlation in case control effects. Also, going back to the earlier results, we can obviously estimate left-right hemispheric differences in the age of the milestones that we derived. Here illustrated for the peak, we see quite substantial timing differences across phenotypes, where blue regions indicate the right side peaking late and left is indicated as red. Now that raises the question, can we not just model asymmetry directly? And indeed, that's what we did next. We estimated the asymmetry index as left minus right over left plus right. And I'm showing you now full lifespan trajectories of asymmetry development from prenatal periods until old age. You see that each of these trajectories is a single normative model for a particular region for each phenotype. Note that anything below 0 indicates right dominance, and anything above 0 indicates left dominance. I've colored these by the rank in asymmetry. I'm going to focus in on cortical thickness for a moment, because you see this beautiful pattern of initial very strong asymmetry in the early life periods that then becomes much less pronounced in later life. It doesn't disappear, but it's less pronounced. Also, you can see a pattern of fronto-occipital left to right dominance if you average across the lifespan. Now, characterizing these regional asymmetry trajectories further, we estimated the area under the curve for each asymmetry trajectory. Now, initially, we did this as the absolute integral, so the absolute difference from 0 on, and here we show this for cortical thickness and gray matter volume, where a dark red value indicates a more asymmetric region, a lighter value is less asymmetric. We can obviously also estimate the signed integral, that's what we did next, where a blue color indicates right dominance and a red color the left dominance. Again, we observe this gradient in cortical thickness with a bit more varied results in gray matter volume. So next, a temporal resolution to these results. I estimated the exact same things I just showed you on the previous slide, but now in age bins or our epochs. And for cortical thickness, we observe this beautiful early development, really strong asymmetry development during this period, and much less pronounced in aging. Again, right dominant is blue, left dominant is red here. I briefly want to highlight the gray matter volume results here. If you focus in on the early childhood, you see really strong leftwards asymmetries in language regions. And that's a really nice validation, I think, because we obviously know that that is a period of language acquisition in children. Now, lastly, and really to summarize the results from the previous slide, I estimated the spatial correlation between epochs of asymmetry. So here I indicate correlation values between each combination of these epochs. And what we observe is two periods of asymmetry development separated by early childhood, so early life, early development, and later life or aging. Now, to recap, we have estimated left and right separate regional trajectories of brain development and characterized them in terms of their milestones of development. We've also estimated asymmetries in these milestones and find quite substantial differences. We next estimated asymmetry more directly and see strong variation in asymmetry over the course of life. We further observed spatial effects of asymmetry development that may relate to the cognitive skills acquisitions in relevant periods of life. Now, collectively, I hope really that these results have demonstrated the potential of mapping asymmetry development over the course of life and for identifying periods of opportunity and vulnerability. And I personally am therefore very excited to next explore more the case control differences in asymmetry centile scores in future work. And with that, I would like to thank my supervisory team and my collaborators. And I will also say that we're currently advertising a postdoc position, so please apply and come to Margaret's poster that is still this afternoon. Thank you.
Thank you very much. We have time for one question. Any questions from the audience? If not,
May I ask a question? Just one quick question. So you showed that the cortical thickness in like BA 44, 45, which is the language region, that's not really a symmetry. So I wonder like if you have any comments, or if, and also showed the result, but I wonder if any sex differences in these regions. Thank you.
So yes, we see sex differences. Sorry, I'm starting from the back here. We see sex differences in general. We're estimating this as an intercept at the moment. In cortical thickness, much less so than in gray matter volume, for example. I think we do see asymmetry effects in BA25, as far as I'm aware. It's just maybe less strong than you see in other areas. I don't think any of that was non-asymmetric, basically.
Thank you.
Thank you.
thank you.